everyone. Me and my group members are from D1 and 1P8. My name is Nurul Ali Nakila binti Zulizam. My group members are
Nursuya binti Muhtar, Nurdiana Lanha binti Rasani,
Nur Hasni binti Azam Mustahal,
Aidanisha Anwar binti Muhammad Manisha Kubis. Today we are going to present about recombinant DNA technology.
Hasni,
yes,
do you know about recombinant DNA technology?
I'm not really aware about this topic.
It's okay, we are going to learn the topic. I'm so excited.
Yes, me too.
Hi students, today we are going to learn about recombinant DNA technology. In this topic, we are going to learn about definition, tools and methods. The definition for recombinant DNA technology is a series of producers that are used to join together recombine DNA segments of two or more different sources in vitro. So the tools are target gene, restriction enzymes, DNA cloning vector, host cell, modifying enzyme. While the method is isolation, clip or cut, insertion, transformation and amplification.
Okay, first I would like to explain about tools that are used in recombinant DNA.
Okay, hi everyone, it's me here, Diana. So the conclusion, I have shown you the slides before. You have know that there are 5 tools are used in recombinant DNA. First is target DNA, the sequence of DNA containing gene of interest, means the gene that we want to copy. Next is restriction enzyme which cuts DNA molecules at the restriction site and produces short restriction fragments, where it will produce either sticky ends, the single-stranded ends, or the blunt ends, where the cut is made between the adjacent C and G. The next tool is DNA cloning vector, where DNA molecule which can carry fragment into a cell and replicate in that cells. And host cell, a cell which receive a recombinant DNA and allows the replicate DNA of recombinant DNA. And lastly, the modifying enzyme, the example of DNA ligase and Taq polymerase. So here, I hope you understand the presentation well.
Okay, to make sure that you guys understand this topic, I have some questions to ask. First, describe the definition and characteristic of cloning vector and host cell. The second one is explain the types of cloning vector. So who wants to answer?
Me. I know the answer for it. The definition and characteristic of a host cell. A host cell is an organism that receives recombinant DNA for the cloning process. Alright, the characteristic of host cell is able to accept the recombinant DNA by transformation. Okay, next. able to maintain the recombinant plasmid from one generation to another. And the last one is able to amplify the gene product of the recombinant DNA.
Okay, so who wants to answer the next question?
Me. Okay, a vector is an agent that carries foreign DNA fragments into a host cell. So there are two types of cloning vector. So the first one is plasmid. A plasmid is a circular DNA molecule which replicates separately from the host chromosome. So the second one is cosmid. A cosmid is a hybrid between a plasmid and a phage lambda chromosome.
So now, I am going to present about the method in gene cloning. There are 5 steps involved in gene cloning, which is isolation, fifth cut, insertion, transformation and amplification, and the last one is screening.
The process of cloning a gene involves insertion of a DNA sequence into a piece of autonomously replicating DNA, such as a plasmid. The transfer of this new plasmid construct into a bacterial cell, and the growth of a population of bacterial cells containing the plasmid. In this way, a clone is generated consisting of many exact copies of the inserted sequence. A plasmid contains a bacterial origin of replication that is required for it to replicate in a bacterial cell. The plasmid also carries a selectable marker gene that makes the bacterial cell transformed with this plasmid, able to grow in a specific defined medium. In our example here, the plasmid contains a gene that makes the bacteria resistant to the antibiotic ampicillin. In another location in the plasmid is a copy of the LAC-Z gene that will produce the enzyme beta-galactosidase when the plasmid is inside the bacterial cell. The LAC-Z gene in the plasmid has a region that is recognized by specific restriction endonucleases. Each restriction endonuclease that recognizes this region cuts the plasmid only in this location, so it is a unique site for that enzyme. Most restriction enzymes cut the 2 DNA strands in different places and thus produce regions of unpaired sequence called overhangs or sticky ends. The piece of DNA we wish to clone must now be generated. In our example here, we will clone the beta-globin gene obtained from human chromosomal DNA. If we treat human chromosomal DNA with the same restriction endonuclease used to open the cloning vector plasmid, a set of specific fragments will be generated that vary in size. Provided that there is not a restriction site inside the beta-globin gene, one of the fragments generated will contain the complete beta-globin gene. However, other fragments of human DNA not containing the beta-globin gene will also be generated. When the fragments of human chromosomal DNA are mixed with the plasmids that have been linearized by the restriction endonuclease, the sticky ends of the plasmid DNA and the human DNA fragments can hybridize with each other, forming double helical structures at the overhangs. These structures can be further stabilized using the enzyme DNA ligase. This enzyme ligates DNA strands by covalently connecting breaks that occur in the DNA backbone. This is exactly the situation at the sites where the double-strand overhangs have now hybridized. DNA ligase closes these strand breaks, making a new circular DNA cloning vector molecule with a human DNA fragment ligated into its unique restriction site. While some plasmids will receive the human DNA fragment containing the beta-globin gene, other plasmids will be generated containing other fragments of human DNA that do not contain the beta-globin gene. Still, other plasmids will simply have sticky ends that find each other and no human DNA will be inserted. DNA ligase will then close these plasmids again, regenerating the original cloning vector. Thus, at the end of the ligation reaction, 3 types of cloning vector plasmids will be formed. The population of various plasmids is then transformed into a population of bacterial cells that have been specially treated to make them competent to receive DNA. This generates a population of transformed bacterial cells which contain cloning vector plasmids with or without human DNA inserts. These cells are then plated onto solid growth media containing ampicillin. Only cells that have taken up a plasmid are able to grow and form bacterial colonies. Petri dishes filled with solid growth media plus ampicillin also contain a colorless substance that the LAC-Z gene protein, namely beta-galactosidase, can convert to a blue substance. After overnight growth, those colonies that arise from cells that received a plasmid without an insert will appear blue. This is because the LAC-Z gene in these plasmids is able to produce a functional beta-galactosidase. In contrast, colonies that received a plasmid with a human DNA insert will be unable to synthesize beta-galactosidase because the human DNA fragment would disrupt the connection between the LAC-Z gene and its promoter. These colonies will appear white. White colonies can then be selected and examined for the presence of human beta-globin gene. A colony containing this gene can then be saved and grown for further study of the cloned beta-globin gene.
I am going to summarize the methods in gene cloning. As shown in the diagram book, the first step is isolation. The bacterial plasmid and the foreign DNA containing the gene of interest will be isolated. The next step is cut or cleave. The isolated bacterial plasmid and the foreign DNA containing the gene of interest are cut with the same restriction enzyme, which will produce sticky ends on both the foreign DNA fragments and the plasmid. Next is insertion. So, the recombinant plasmid are joined with DNA ligase. Next step is transformation, which is the introduction of cloning vector into bacterial cells, which is host cell. This will allow the recombinant plasmid to multiply. The next step is amplification. Amplification will allow bacterium to reproduce. As the bacteria reproduce, the recombinant DNA is also replicated or cloned. So, the last step of gene cloning is screening. Screening is used to identify bacteria clone containing recombinant DNA. As an example, blue-white screening. So, blue-white screening is when the bacteria is plated on agar containing antibiotic, ampicillin and sugar at skull. Then it is incubated until colonies grow. So, to make sure you guys understand the method well, I have a few questions. Can any of you describe the steps in gene cloning?
I think I can answer the question for A. The step for number 1 is isolation. Isolate the plasmid from the bacteria and isolate the target DNA from donor cells. Next is cutting. Cut donor DNA and plasmid DNA using the same restriction enzyme. The restriction enzyme cuts the DNA of interest and the plasmid only a specific nucleotide sequence. The ends of the cut have an overhanging piece of single-stranded DNA called sticky ends. The next step is insertion. Gene of interest is inserted into the plasmid which has complementary sticky ends. DNA ligase joins the complementary sticky ends, forming the recombinant DNA. The next step is transformation and amplification. Recombinant DNA is transferred into the host cell by transformation process for cloning purpose or amplifying gene of interest. The last step is screening. Identify clones carrying gene of interest.
Good. Next question. Can anyone explain the screening process in gene cloning? You, Hasnin, would you like to try?
Sure. Screening. The process of sorting through thousands of bacterial colonies with foreign DNA to find bacteria containing gene of interest. Only bacteria that have the ampicillin-resistant plasmid will grow, and the blue colony carries the non-recombinant plasmid with intact LAC-Z. The white colony carries the recombinant plasmid with disrupt LAC-Z.